Previously, we have studied about the glycolysis, which was the partial oxidation of glucose molecule into a 3-carbon molecule that is called the pyruvate. And that glycolysis that was we can say that was divided into 10 chemical reactions, and in those 10 chemical reactions, 3 chemical reactions they were irreversible, while the 7 chemical reactions they were reversible. And now we will discuss about the regulation of this pathway, the glycolysis, regulation of glycolysis. So, regulation of glycolysis. As you know, the chemical reactions of glycolysis, these are like glucose, from glucose to glucose 6-phosphate, and from here fructose 6-phosphate, and from here fructose 1,6-bisphosphate, then comes dihydroxyacetone phosphate, as well as glyceraldehyde 3-phosphate. Then this dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate, and then this reaction goes on, and we have 1,3-BPG, and then we have 2-PG from 3-PG. First from this reaction, we will have 3-PG, 3-phosphoglycerate, which means then we will have 2-PG, and then there will be phosphoenol pyruvate, and at the end we will have pyruvate. So these were the chemical reactions of the glycolytic pathway. And today, as we are going to discuss about the regulation of glycolysis, so remember, those chemical reactions, they will be the regulatory chemical reactions which are irreversible. So from here, this one, this is the irreversible chemical reaction, as well as this one, this is also an irreversible chemical reaction, and this one, this is also an irreversible chemical reaction. If we number these chemical reactions, this is chemical reaction number 1, 2, 3, 4, 5, and this one is chemical reaction number 6, 7, 8, 9, and 10. And in these chemical reactions, which one are the irreversible chemical reactions? The reaction chemical reaction number 1, let me write it over here, from glucose to glucose 6-phosphate. This is the chemical reaction number 1 of glycolysis. And then if you look over here, the chemical reaction number 3, this is also irreversible chemical reaction. So fructose 6-phosphate to fructose 1,6-bisphosphate. This is the chemical reaction number 3, and then the next irreversible chemical reaction is reaction number 10. So this reaction number 10 is phosphoenol pyruvate to pyruvate. So whenever you are being asked that write down the regulation of glycolysis or how this regulation is being regulated, so always remember that all the chemical reactions which are irreversible in the glycolytic pathway, those chemical reactions, they will be the regulatory chemical reactions of this pathway. And what are those regulatory chemical reactions? Chemical reaction number 1, this is a regulatory chemical reaction. Chemical reaction number 3, this is also a regulatory chemical reaction. And reaction number 10. This is also a regulatory chemical reaction. In few books, you will find total 10 chemical reactions, and in few books, there will be written total 9 chemical reactions. Because in those books, they are counting this chemical reaction, these 2 chemical reactions, as a single chemical reaction. So that's why in few books you will find 9 chemical reactions, and in few books you will find 10 chemical reactions. An easy trick to remember the regulatory steps of this glycolytic pathway is simply remember the chemical reaction number 1, 3, and last. These are the regulatory chemical reactions. Once again, I am repeating that chemical reaction number 1, chemical reaction number 3, and the last chemical reaction, whatever it is, either it is the 10th chemical reaction or 9th chemical reaction. These are the regulatory chemical reactions. And these chemical reactions, these are further regulated by their respective enzymes. Like, for example, in first chemical reaction, the enzymes which are responsible, these enzymes are Glucokinase or Hexokinase. Let me first explain this regulatory step. If we talk about the glucokinase, first of all, always remember that this glucokinase is a glucose sensor for the homeostasis of glucose. Once again, I am repeating that this glucokinase, this will act as a glucose sensor for the carbohydrate diet or for the glucose levels, which means whenever there is a rise of glucose levels in the blood, at that time this sensor will detect that those increased level of glucose, and as a result it will start the glycolysis. That's why this glucokinase is a very important enzyme, and how it is involved in the regulation of this regulatory step. Whenever, remember, this glucokinase, it has its active form as well as its inactive form. When we talk about the inactive form, it means in the nucleus, this is the nucleus. This glucokinase is attached with our regulatory protein inside the nucleus. And what is that protein? That protein is, suppose we say that this is the regulatory protein and this is the enzyme. This regulatory protein, it is named as glucokinase regulatory protein. This is the regulatory protein, and this is the enzyme glucokinase. This is the enzyme, and this whole portion is the nucleus. This is the nucleus, inside of the nucleus. So, what is going to take place, that whenever there will be an increased level of glucose in the blood, suppose this is the glucose, and when this glucose level is increased, then what will happen? As a result of this increased glucose level, this association, this will break. And when this association will break, the glucokinase, it will be released into the cytosol. When this glucokinase is released into the cytosol, then this glucokinase will become active and it will phosphorylate the glucose into glucose 6-phosphate. And whenever the levels of fructose 6-phosphate increases or the glucose level in the blood that is lower, at that time this glucokinase, it will go back to the nucleus and it will bind with the glucokinase regulatory protein. Like this, for example, when we have decreased level of glucose, as well as when we have increased level of fructose 6-phosphate. As a result of this, they will trigger the glucokinase, and this glucokinase, it will go back to the nucleus, and again it will become inactive in the nucleus. So once again I am going to repeat what I have explained you about the first regulatory step, that in the first regulatory step we have two enzymes, glucokinase as well as hexokinase. Hexokinase, it will work in all conditions, because if there is a decreased level, at that time we need the energy, at that time it will always be working, and it is we can say that it is always phosphorylating the glucose into the glucose 6-phosphate. Either we need energy or we don't need energy. If we need energy, then it will continue, this glucose 6-phosphate will continue to shunt itself into the catabolic pathways, and if we don't need energy, it will shunt itself into other pathways like the synthesis of glucose or gluconeogenesis. So that's why this enzyme hexokinase, this is not so much significant as compared to this glucokinase. And what is the significance of this glucokinase? This glucokinase, it is a glucose sensor. Whenever we have taken our rich carbohydrate meal, at that time, what will happen, that this increased level of glucose, these will be detected by the glucokinase. And this glucokinase, what it will do, simply it will phosphorylate this glucose into glucose 6-phosphate. And question mark over here is that how this glucokinase, it becomes active. And the answer to this question is, this glucokinase, it is always present whenever we are talking about its inactive form. That means in the inactive form, it is always present in the nucleus with a regulatory protein, which we are naming as glucokinase regulatory protein. And in the nucleus, when it is with the association of this regulatory protein, it is inactive. And when it has to be active, then there must be the increased level of glucose, and as a result of this increased level of glucose, this glucokinase, it will dissociate itself from the regulatory protein, and it will come into the cytosol. When it will come into the cytosol, it will simply phosphorylate the glucose into glucose 6-phosphate. And when the glucose levels, they are back to the normal level, they are back to the normal state, they are not so much high or they are not so much increased, at that time what will happen, that those decreased level of glucose or the increased level of fructose 6-phosphate. These levels are increased by the increased phosphorylation of this glucose. When this glucokinase, it will become active, it will phosphorylate more and more glucose. And when it is phosphorylating more and more glucose, it means we are having more glucose 6-phosphate. And when we are having more glucose 6-phosphate, it is being converted into more number of fructose 6-phosphate. So as a result of the increased level of fructose 6-phosphate or decreased level of glucose, this glucokinase, it will go back to the nucleus, and again it will bind with the glucokinase regulatory protein. So this is the first regulatory step of the glycolysis. And this glucokinase as well as hexokinase, it can also be controlled with the help of hormones. Not only the level of glucose, they are regulating or affecting this glucokinase or hexokinase. These can also be regulated with the help of insulin or glucagon. And here we are discussing about the utilization of glucose. So it means insulin is responsible for the activation of this glucokinase enzyme or the activation of hexokinase enzyme. And glucagon, what about the glucagon? Glucagon simply, it inhibits the glucokinase as well as the hexokinase activity. So this is the first regulatory step. And next comes the second regulatory step. In the second regulatory step, this is very, very important step because this step is also referred as rate limiting step. This chemical reaction, this is also referred as rate limiting step, because this reaction will decide that either the pathway will be glycolytic or the pathway will be gluconeogenic. Let me explain this. The regulatory enzyme involved for this chemical reaction, that will be phosphofructokinase 1. This phosphofructokinase 1, it can be regulated allosterically as well as the hormonally. So let me explain it over here. This phosphofructokinase 1, this is also a regulatory enzyme, and this chemical reaction is also named as rate limiting step because this reaction is responsible for the decision, for making decision that either this reaction, this pathway, it will go toward the glycolysis or toward the gluconeogenesis. So that's why we name this chemical reaction as a rate limiting step. And this PFK1, the activity of the PFK1, it is regulated at two levels, PFK1. Number one, allosterically, and number two, by the levels of fructose 2,6 base phosphate. First, let me explain this. Allosterically, as you know very well, that allosteric mean whenever a substrate or an inhibitor or an activator, it binds with the enzyme other than active site. Such type of activity or such type of substrates or compounds, they are called as allosteric compounds, and such type of the regulation is called as allosteric regulation. And over here, the allosterically, it is regulated either by the levels of ATP or by the levels of citrate. These two compounds, these are responsible for the allosteric, either upregulation of the phosphofructokinase 1 or the downregulation of phosphofructokinase 1. But now the question is how? Whenever there is the increased level of ATP, it means our body, our cell is sufficient with the energy. It does not need any extra energy. So at that time, these increased levels of ATP, they simply inhibit the activity of phosphofructokinase 1. So what we are seeing that this increased level of ATP, it will inhibit the enzyme. So second one, and whenever there is a decreased level of ATP, or in other words, you can say that increased level of AMP. At that point, in increased level of AMP or decreased level of ATP, that will activate the phosphofructokinase 1 enzyme, and the reaction will proceed toward the formation of pyruvate, and that this pathway then at that point it will yield the energy. So second one is the citrate. As we know very well that this citrate, this is a very important, we can say that important pathway, important point for the regulation of this enzyme. This citrate, this is the product of TCA cycle. We have already studied this, that what is the TCA cycle. TCA cycle is a tricarboxylic acid cycle, and it is the major pathway or the last final pathway for all the intermediates, or either those are the carbohydrate compounds or the non-carbohydrate compounds which are responsible for the energy production. Only I am talking about those compounds. And this TCA cycle is the final pathway for all those intermediate compounds which are being produced by the metabolism of either amino acids or by the protein, by the lipids or by the carbohydrates. And after this pathway, we get the final amount of energy, final budget of energy. So this citrate is the compound of this TCA cycle. Whenever there are increased level of citrate, whenever there are the increased level of citrate, what it will simply do, it will inhibit the activity of phosphofructokinase 1, because the increased level, they will show that it is exceeding from the capacity of TCA cycle, and it will simply signal to the phosphofructokinase 1 that please stop the activity. And this phosphofructokinase 1, it will be inhibited, or it will stop the activity, or it will stop the phosphorylation of this fructose 6-phosphate to fructose 1,6-bisphosphate. And the second one is in the regulation of this enzyme activity, that is the levels of fructose 2,6-bisphosphate. This fructose 2,6-bisphosphate is a potent activator of this enzyme, and it is a potent inhibitor of the gluconeogenic enzyme at this point. So this one is very important, and the levels of fructose 2,6-bisphosphate or the fructose 2,6-bisphosphate, this will work either the ATP levels are lower or the ATP levels are higher. So this will decide that either the pathway, it will proceed toward the formation of pyruvate or toward the formation or synthesis of glucose through gluconeogenesis. And with on this basis, on the basis of these levels, we decide or we say that this chemical reaction is a rate limiting step. So but the question is how the fructose 2,6-bisphosphate, these molecules are produced or these are synthesized. The answer to this is fructose 2,6-bisphosphate, it is synthesized from the fructose 6-phosphate with the help of an enzyme which we name as phosphofructokinase 1. So let me explain this. What we are talking about, the levels of fructose 2,6-bisphosphate. And this fructose 2,6-bisphosphate, it is produced by or from fructose 6-phosphate to with the help of enzyme phosphofructokinase 2. Last time I have explained you that phosphofructokinase 1 means the phosphorylation of fructose molecule at the carbon number 1. So that's why it was named as phosphofructokinase 1. And over here, phosphofructokinase 2 means the phosphorylation of this molecule at the carbon number 2. And it is named as fructose 2,6-bisphosphate. And this enzyme, this PFK2, this enzyme has two domains. One is kinase domain and second is phosphatase domain. So this kinase domain, we can say that it is used for the phosphorylation. And whenever there is the phosphorylation, this enzyme, it will become inactive. And phosphatase domain, it means dephosphorylation. And whenever there is dephosphorylation, it means it will become active. And when this enzyme will become active, what it will do? It will simply phosphorylate the fructose 6-phosphate into fructose 2,6-bisphosphate. And when the levels of this fructose 2,6-bisphosphate, they will increase. These increased levels then activate this phosphofructokinase 1. And as a result, this reaction will move forward. And the glycolytic pathway, it will start. And question here is that how these two domains are being regulated. So answer to this is, let me explain this with the help of a simple diagram at this. For example, this is the cell membrane. And we know very well that in the cell membrane, we have, for example, receptors. There will be two hormones. Number one, that will be the insulin hormone, and number two, that will be the glucagon hormone. And when we talk about the insulin, it means insulin is helping us in lowering the glucose levels. And how it is helping us in lowering of glucose levels? Simply by increasing the utilization of those glucose molecules. At this point, as we are discussing about the activation of this PFK1, what will happen? That the insulin hormone that will come, suppose we say that this is the insulin hormone. This is insulin hormone, suppose. When it will become attached to this receptor, there will be a signal cascade. There will be a signal cascade. As a result of that signal cascade, or as a result of that signaling, what will happen that the phosphatase domain of phosphofructokinase 2, it will become active, like this. Suppose this is the phosphofructokinase 2 enzyme, and as a result of when the insulin comes, it binds with the receptor, it will send signal towards the phosphofructokinase 2, and in the phosphofructokinase 2 we have two domains. Number one is phosphatase domain and number two is the kinase domain. The phosphatase domain will become active and it will dephosphorylate this enzyme. When this phosphofructokinase 2, it will become dephosphorylated, whenever it is in dephosphorylated form, this will become active. And when this enzyme is active, it simply starts the formation of fructose 2,6-bisphosphate. So here the synthesis will start. So when the synthesis of this fructose 2,6-bisphosphate, its synthesis will start. As a result of this, the levels will rise, and these increased levels, they will activate our concerned enzyme, which is PFK1. When this PFK1, it is active, then we will have glycolysis. So this is the pathway through which this activity of PFK1 is being regulated. And when there comes the glucagon, that glucagon hormone, when it will become attached to the receptor, and that glucagon hormone will activate the kinase domain rather than the phosphatase domain. And as a result of the activation of this kinase domain, this PFK2, it will be phosphorylated. And when it is phosphorylated, it will become inactive rather than being active. And when it is inactive, there will be no synthesis of fructose 2,6-bisphosphate. And when there is no synthesis of fructose 2,6-bisphosphate, the level will drop. and this enzyme will become inactive, and those dropped levels of this fructose 2,6-bisphosphate, they will activate another enzyme which is fructose 1,6-bisphosphatase, and this is the enzyme of gluconeogenesis. And as a result of this cascade or activation and inactivation, there will be no glycolysis and there will only be the gluconeogenesis. So that's why we say that these gluconeogenesis and glycolysis, these are the reciprocal pathways, because at the same time only one reaction or one pathway that will operate. Either that pathway can be glycolysis or it can be the gluconeogenesis. Once again, I'm going to repeat about the regulation of activity of PFK2, that how it is being regulated and how it is going to affect the activity of PFK1 enzyme. Simple, we have two hormones, insulin and glucagon. Whenever the glucose level it will rise or it will increase in the blood, there comes the insulin. And whenever there is a release of insulin, it means now body has to utilize the glucose. And this insulin, it will become attached to the receptor, and after being attached to this receptor, there will be a signal cascade till the PFK2, phosphofructokinase 2. And this phosphofructokinase 2, as I have explained you that, it has two domains. Either number one is kinase domain and number two is phosphatase domain. Kinase domain is for the phosphorylation, and whenever there is the phosphorylation, that PFK2 it will become inactive. And phosphatase domain, that is for the dephosphorylation, and whenever there is the dephosphorylation, it will become active. And with the help of this insulin, the phosphatase domain of PFK2 that will become active and it will dephosphorylate this enzyme. When this enzyme it will be it is dephosphorylated, simply it becomes active. And when this enzyme becomes active, it starts synthesizing the fructose 2,6-bisphosphate. And as a result of this, there will come a point at which the fructose levels of fructose 2,6-bisphosphate, they will increase. And those increased level of fructose 2,6-bisphosphate, they will activate the phosphofructokinase 1. When this phosphofructokinase 1, it will become active, the glycolysis will start. And in case of glucagon, it is opposite. Like how? When the glucagon, it will come and it will bind to the receptor. After the binding, there will be a signal cascade, and the kinase domain that will become active. And that kinase domain that will phosphorylate this enzyme, and after the phosphorylation of this enzyme, it will become inactive rather than being active. And when it is inactive, there will be no synthesis of fructose 2,6-bisphosphate, and when there is no synthesis of this molecule, this will become inactive or it will be inhibited. When it is inhibited, there will be no glycolysis. And if no glycolysis, then what happened? Then there will be the activation of another enzyme which we name as fructose 1,6-bisphosphatase. And after the activation of that enzyme, there will start the gluconeogenesis. So this was for the regulation at the third chemical reaction with the help of this phosphofructokinase 1. Then comes the next chemical reaction, which is the phosphoenol pyruvate. In the next chemical reaction, in the phosphoenol pyruvate, the major enzyme which is involved, that enzyme is named as pyruvate kinase. Again, this is a very interesting enzyme, and this enzyme is clinically very important. Now let me explain this. Pyruvate kinase. Again, this pyruvate kinase, this enzyme, it also has two domains. Number one is kinase domain and number two is phosphatase domain. Or let me make it simple for you. That this pyruvate kinase, it exists in two forms. Number one is phosphorylated form and number two is dephosphorylated form. This dephosphorylated form, this is inactive form, while this dephosphorylated form is active form, not the inactive. This dephosphorylated form is the active form, while this phosphorylated form, it is inactive form. The activity of this enzyme, it is regulated with the help of hormones, or we can say that it is regulated by the covalent phosphorylation and dephosphorylation. And the major role for the activity or for the activation or inactivation of this enzyme is the insulin hormone as well as the glucagon hormone. These two hormones, these are responsible for the activation or inactivation of this pyruvate kinase, as well as it is also allosterically regulated with the help of levels of AMP, increased levels of AMP. So we can say that the activity of this enzyme is either hormonally regulated or allosterically regulated. The question is how? Let me show this to you with the help of diagram. For example, again, this is the cell membrane. In the cell membrane, for example, we have a receptor. Whenever we need to inactivate or activate this enzyme, there will come a hormone. And in this case, in case of carbohydrate metabolism, the major role of the hormones which are involved, those are either epinephrine or glucagon or insulin. Insulin is for the lowering of glucose levels, and glucagon or epinephrine is to increase the level of glucose. And whenever we have to lower down the levels of glucose, we have to utilize that glucose. And whenever we have to increase the level of glucose, then we don't need to utilize that glucose, but we need to synthesize the new glucose. So this is the difference between the action of insulin and glucagon. And over here in this example, as I'm going to explain you about the pyruvate kinase, either it exists in the phosphorylated form or the dephosphorylated form. Phosphorylated is the inactive form and dephosphorylated is the active form. When there comes the insulin, for example, this is insulin. When there comes the insulin, this can be any receptor like this. There will be a signal cascade. And as a result of that signaling cascade, there will be the activation of another enzyme which is very important, that is protein phosphatase. This protein phosphatase, what it will do, it will attack or it will we can say that it will affect the pyruvate kinase enzyme. When it will affect the pyruvate kinase enzyme, then this pyruvate kinase enzyme, it will become inactive. Question is, sorry, it will become active. Let me repeat it. When there comes the insulin, this insulin, it will bind with the receptor. When it will bind with the receptor, there will be a signal cascade in the cytosol, and the protein phosphatase, this enzyme, it will become active. And what it will do, it will simply dephosphorylate. What it will do, it will simply dephosphorylate this enzyme. When this protein phosphatase will dephosphorylate this enzyme, as a result of this dephosphorylation, this enzyme, it will become active. And when this enzyme will become active, there will be the formation of pyruvate molecule. And in second case, whenever the receptor is glucagon, sorry, whenever the hormone is glucagon, not the receptor. Again, this is the receptor, and here is the hormone. Suppose this is the glucagon hormone. This glucagon, it will become attached to this receptor, and after being attached to this receptor, there will be the formation of cyclic AMP molecules through another receptor which we name as adenylate cyclase with the help of G protein. What happened? That when there comes the glucagon, this glucagon will become attached to the receptor. This receptor, with the help of G protein, it will activate the adenylate cyclase, and this adenylate cyclase will start forming cyclic AMP. And this cyclic AMP, with the help of signal cascade, with the help of protein kinases, it will attack on pyruvate kinase molecule. This is the pyruvate kinase. And what it will do, it will simply phosphorylate. What is it doing? It simply phosphorylate this pyruvate kinase. And when this pyruvate kinase is phosphorylated, it will become inactive. And when it will become inactive, there will be no glycolysis. Let me explain this to you once again, that we have two hormones. Number one is insulin and number two is glucagon. And how these are going to affect to upregulate or downregulate the activity of this pyruvate kinase enzyme, that when there comes the insulin hormone, it will become attached to the receptor. After being attached to the receptor, there will be a signal cascade toward the protein phosphatase enzyme, and this protein phosphatase enzyme, it will simply dephosphorylate the pyruvate kinase. And when there is the dephosphorylation of this pyruvate kinase, it will become active, because I have told you that the dephosphorylated form is the active form. And when this enzyme will become active, it will form the pyruvate molecule along with the two molecules of ATP, because we have two molecules of phosphoenol pyruvate and two molecules of pyruvate. But when comes the glucagon or epinephrine, which means we are already have less number of glucose molecules in our blood, or the lower concentration of glucose in our blood, at that time we need to increase the glucose concentration. So at this point, this glucagon, it will become attached to the receptor, and with the help of this receptor and with the help of G protein, it will activate another receptor, adenylate cyclase. This adenylate cyclase will form cyclic AMP, and this cyclic AMP, it will activate another enzyme which we are naming as protein kinase. When this protein kinase is active, it will phosphorylate this key enzyme. When this key enzyme is phosphorylated, it means this is now inactive and there will be no formation of pyruvate. Rather than, body will start converting this phosphanol pyruvate back into 2-phosphoglycerate as well as till the glucose. So this was the regulation of glycolysis. And the last thing about the pyruvate kinase, that this pyruvate kinase is very significant. Why significant? Because it is clinically, it can cause the anemia. That we will discuss in next lecture.